Dear students and viewers, Assalamu alaikum. So today we will be teaching you principles of penicillins pharmacotherapy with emphasis on antibacterial drugs. Uh, why there is a need for antibiotic therapy? Because nowadays chances of infections are very high and antibiotics are mostly prescribed. So that necessitates an urgent need for the rational use of antibiotics. Some facts about bacteria, and bacteria can be classified by shape, like whether they are cocci, bacilli, spirochetes, may be classified on the basis of cell wall, whether that is present or absent, like mycoplasma have cell wall, don't have cell wall, and the other have cell wall, or gram positive or gram negative, based on the thickness of the layer of peptidoglycan layer, and growth requirements, like whether bacteria may be aerobic or anaerobics, or survive extracellularly or intracellularly, or as the case may be. Now recall your secondary school level, where the major components of bacterium were cell wall, cell membrane, cytoplasm, and nucleoid materials. Now, some basic concepts that are very necessary to proceed further, that is MIC and MBC. MIC stands for minimum inhibitory concentration, and MBC stands for minimum bactericidal concentration. Potency is one over the reciprocal of MIC or MBC, so that refers to the potency, and spectrum means that types of microorganisms being covered by a particular antibiotics. Thus, this spectrum determines the utility or the use of an antibiotics in a particular clinical condition that ultimately take as indication or therapeutic uses. And then resistance. Resistance is a global phenomenon because of the misuse of antibiotics, particularly in veterinary and other poultries, when antibiotics are most frequently used. So just for prophylaxis purposes, so this exposes the bacteria of the environment, and thus bacteria try to cope up with the presently available antibiotics. So thus they fight a survival for the fittest. So keep yourself updated with this terminology before we proceed. Then this diagram is I have taken from a book, and it determines that some bacteria, as I told you earlier, are classified as gram positive and gram negative. Gram positive have very thick wall of peptidoglycan, whereas gram negative have a very relatively less thick wall. You see, this is less thick wall, but most important is that the outer membrane is lipopolysaccharide, and proteinaceous bonds and material are there. So this determines that mostly once in the case of gram negative, so this barrier determines the penetration of antibiotic inside into the particular microorganisms. So thus gram negative are naturally built in with a natural barrier to hydrophilic antibiotic, as this is lipophilic, so like dissolve in likes, hence hydrophilic antibiotics cannot penetrate through this into inside the particular microorganisms. Thus this gram positive or gram negative terminology is very important while determining its clinical uses. Clinical uses of antibiotics. These are other possible site of actions, like cell wall synthesis inhibited by cephalosporins, penicillins, bactericins, and there is, you see, DNA agarase. Quinolones are inhibiting are acting at this side rather. So the supercoils this DNA agarase, therefore it will not be open, and once this double helix is not open, so transcription won't be possible. And then the drugs that are going to affect transcription, like RNA polymerases, rapamycins, and then you see the cytoplasmic membrane damaging drugs like polymyxins, and then the protein synthesis inhibitor, whether that is a 30S or 50S, like these classes, pectinomycin, tetracyclines, and at the 50S you see chloramphenicol, macrolides, some macrolide like mycins, and lincomycin and clindamycin. So this determines the site of action. But more important is regarding penicillins. Now I have made a typical bacterium, you see, and for simplicity, another to differentiate from the cytoplasm. So I use a nuclear membrane here, you see, that with the red one. So I take it as a nucleus just for understanding, though the bacteria have nucleoid materials. Now this is a cell wall, you see, and this cell wall I have left blank because this will, so that the ultimate diagram should not be more dense like as it is here. Now what happens when antibiotics, penicillins are given? But you see, for better understanding of the mode of action of penicillins, let we understand some possible simple processes. And that is, you see, they are in the cytoplasm. There is transpeptidase enzyme. I represented that TP, and here it is represented as TP. Here it's a TP. And then you see there are ribosomes, which we are not concerned here with, as we are not, our main focus is not on the protein synthesis in this presentation. So what happens? This transpeptidase enzyme usually incorporates glucose sugars, amino acids, and lipids, and then the cross-linking of this peptidoglycan layer at the level of cell wall is being supported by the transpeptidase enzyme, as the name indicates. This is transpeptidase enzyme. So once the bacteria grows from inside, so likely the cell wall grows as well. Now in the presence of cell wall synthesis inhibitors like amoxicillin, so the amoxicillin first penetrates into the internal of this bacterium, and it is now coming here and act at the penicillin protein binding site. These are PBPS. These are very red on this transpeptidase enzyme. I have tagged it as a red caps, you see, present here also. And once amoxicillin or penicillin, this is called, why it is called as penicillin binding proteins. These are called penicillin binding proteins because penicillins comes here. These are protein in nature and that attach hairs. So what it does, it prevents this enzyme, rather the transpeptidase enzyme, from incorporating the glucose, amino acid, lipids, and the cross-linking of this peptidoglycan layer. So in the presence of amoxicillin here, you see this cell wall cross-linking will not happen, and once it will not happen, so the cell wall synthesis is arrested, is stopped. But the bacteria grows from the from the internal point of view. So you see, internal material grows, and once the internal material grows, so the osmotic pressure inside the bacterium is increased. Since it is stopped here in the presence of amoxicillin, so this side is now weakened. That's I have used this crossword. Now this is weakened, so the bacteria bursts from here, and once it bursts from here, so its contents oozes out, and once its contents oozes out, so this leads to the sedibility or killing phenomena of all penicillins like amoxicillin, ampicillins, tecorcillins, or whatever the classification you like, benzyl penicillin, penicillin V, phenoxymethyl penicillin, cloxacillin, dicloxacillin. So all these have similar mode of action. Now, but bacteria are also clever as well. They pose resistance to this particular antibiotics, penicillins, and once they are going to pose resistance. So how they are going to pose resistance? For that we have to understand certain points as well. In the cell wall there are purines, you see, and these purines, this amoxicillin is influxed, and once it is influxed, it's coming and working here at its transpeptidase enzyme at binding at insulin protein binding site. But there are efflux genes as well, which I have not drawn here, and these efflux genes try to get rid of from this amoxicillin, which is have now arrived at the internal environment. So it is effluxed out. So now amoxicillin come here. So this is one mode of resistance, that there is efflux mechanism is there. You see, efflux mechanism is there, and there is the changes in this purine, the three-dimensional structure, the conformational structure of this purine has been changed. Therefore, the penetration into internal environment of the bacterium won't happens. So this poses another kind of resistance. So there is decreased penetration, and this kind of resistance is usually posed to aminoglycosides like kanamycin, tobramycin, amikacin, and streptomycin class. Changes in this caps-like structure, as I referred to earlier, that is penicillin binding proteins, and it is usually posed by Staph aureus and Streptococcus pneumoniae, and some Klebsiella species like Klebsiella pneumoniae as well, for the extended spectrum beta-lactamases. So what happens? Changes in the penicillin binding proteins also happen. Now the amoxicillin or the penicillin won't be able to attach here properly. Thus, this is another mode of resistance. And as I said earlier, efflux mechanism is there, so it is posed to tetracyclines and macrolides. But very cleverly, the bacteria also synthesizes certain enzymes, and that are secreted through these purines. You see these purines, and these are called beta-lactamases. Since penicillin have beta-lactam ring from chemistry point of view, so they are called as beta-lactamases. And initially they were called as penicillinases because penicillin was the oldest drugs, and it was discovered by Dr. Alexander Fleming, you see. So the enzyme responsible for the degradation for the penicillin were penicillinases, and for the cephalosporinases. But later on, as the chemical structure of both cephalosporin and penicillins carries beta-lactam ring, therefore we call this enzyme as beta-lactamases. Now these beta-lactamases are secreted, and before reaching this antibiotic inside the cell, it is hereby inactivated, hydrolyzed by these beta-lactamases. But the scientists are also clever like bacteria, rather more clever than bacteria, and that is they added a similar structure to amoxicillin or penicillins, having similar structure like beta-lactam, and that is clavulanic acids. You see, so if you add amoxicillin to amoxicillin, the clavulanic acid, you see, this constitutes the augmentin. The scientists are very clever. Their name is augmentin. Augmentin. The activity of amoxicillin is augmented by this clavulanic acid. How it is augmented? That the beta-lactamase cannot differentiate between the clavulanic acid and the amoxicillin, as both of them having the beta-lactam like rings. And now once they have beta-lactam ring, you see, so it have different bonds, so it is not easily attacked. Is it easily attacked? So thus the beta-lactamases are engaged by the clavulanic acids, leaving spared the amoxicillin, and then thus the amoxicillin goes inside. Thus the pharmacological responses are promoted. So that we call that augmentin, and this is available in the market. This is market or trade name, and amoxicillin is with amoxil. Now we added to ampicillin sulbactam with a similar principle, and piperacillin, tazobactam, and tacrolimus plus sulbactam. But the point is that these three combinations are given intravenously, whereas clavulanic acid plus amoxicillin can be given orally. as well, in addition to the parental therapy. So this is the advantage of clavulanic, and perhaps this is very branded drug and this is available in the market. So now you understand the mode of resistance. There are 4 types of mode of resistance, and this inactivation by the enzyme, you see, is these beta-lactamases, and then their hyperproduction of beta-lactamases. So you know that is why the scientists now increase the dose of augmentin, and now 1 gram BD is available in the market. Once we were in the old days, it was 1 gram preparation was not available. So the cell wall synthesis inhibitor bursts the bacteria where its cross-linking of peptidoglycan layer is inhibited. But another point, that is, they are very safe in pregnancy because mammalian cells do not have cell wall, and whereas the bacteria have cell wall, because basically they are plant origins. So this principle makes them more vulnerable towards this cell wall synthesis inhibitors, and this principle makes it more safer in pregnancy because mammalian cells do not have cell wall. But yet in this modern era, the evidence-based practice of medicine necessitates for studies to determine its safety in pregnancy, so they are relatively safe. Now indications: it is used in otitis externa. Penicillins are used in otitis externa. It is gram positive, and it is also used in otitis media. It is also used in pharyngotonsillitis. It is also used in tracheitis, in pneumonia, whether that is bronchopneumonia, lobar pneumonia, and bacterial endocarditis. You see, it's also used in acute maxillary sinusitis and genitourinary tract infection in combination, triple regimen with Helicobacter pylori, for eradication of Helicobacter pylori, where amoxicillin 1 gram BD is used for 10 to 14 days, along with omeprazole 20 milligram and plazomicin 500 milligram. So possible side effects and ADR, that is increasing the GPT level, skin rashes, and drug allergy. So where penicillin therapy fails, when there are atypical microorganisms like mycoplasma, usually Mycoplasma hominis, you see, that is mostly involved in pelvic inflammatory diseases in female. So this risk of failure of therapy is there. Atypical pneumonia and some microorganisms that do survive intracellularly. As side effects of beta-lactam and penicillins, you see rash, as I said earlier, anaphylaxis, serum sickness, diarrhea, hemolytic anemia, CNS toxicity, seizure, and that usually occur in higher doses. But very common is skin rashes, anaphylaxis. Therefore, they are used after test dose. I hope now you understand. Thank you.